You are looking at two mice born on the same day in the same litter. Both were genetically altered to have a gene that accelerates the rate of DNA breakage and repair, a process that corrects the mutations that constantly occur in DNA. In the older-looking mouse, this altered gene is active. In the younger mouse, it's inactive. The older-looking mouse underwent epigenetic changes associated with aging because of this accelerated break and repair process. Essentially, this showed researchers at the Harvard Medical School that it may be the wear and tear on our DNA that causes us to physically age as we get chronologically older. If we can use this process to accelerate aging, can we find a way to safely slow it down? Extending the human lifespan is nothing new, but the elusive goal of living healthier for longer has been far more challenging. Holding on to our youth has always been a tantalizing prospect, but we may not have to settle for sports cars much longer. A handful of recent breakthroughs, from unexpected drug effects to genetic therapy, have some experts in the longevity field projecting that we may see the fountain of youth within the next few decades. So is it possible to decouple biological age from years lived? For centuries, humans have strived to extend their lifespans, and the progress has been astounding. Thanks to medical advancements, life expectancy has doubled over the years, and we now have a chance to live twice as long as our ancestors did. However, this remarkable achievement has led to a new challenge: aging itself. Today, aging is the leading cause of over 2/3 of global deaths, with more than 100,000 people succumbing to its effects every day. As we age, we become more vulnerable to various age-related ailments, including cancer, Alzheimer's disease, and mobility issues. The sad reality is that though we've extended our average lifespans, we have made less progress on improving healthy function in the additional years we've gained. Yet, hope is on the horizon. Aging. A complex biological process involves a myriad of factors and cellular changes that contribute to the decline of our bodies over time. Various longevity research teams are dedicated to uncovering ways to slow down or even reverse these aging processes, and their approaches are as diverse as they are innovative. One approach gaining significant attention focuses on senescent cells, malfunctioning cells that accumulate as we age. Like other cells, senescent cells multiply, but unlike other cells, they don't die off when they stop multiplying. These cells have been linked to a range of age-related ailments, including cancer, Alzheimer's disease, and declines in eyesight and mobility. Scientists are analyzing vast amounts of data, often using AI algorithms to identify chemical compounds that can effectively eliminate senescent cells. By targeting these cells, researchers aim to prevent or reverse their detrimental effects on the aging process. One such compound came from a rather unexpected source. Metformin was developed to manage blood sugar levels for individuals with diabetes. However, it fell out of favor due to negative side effects compared to insulin, such as gastrointestinal upset and lactic acidosis. In recent years, researchers have been investigating its potential as an anti-aging drug. This effect was noted in a study that compared the lifespans of two groups of people with type 2 diabetes. One treated with metformin, the other with sulfonylurea, each matched with non-diabetic control groups. They were surprised to find the group using metformin had the longest lifespans, longer even than the control groups without diabetes. Preliminary evidence from animal and observational studies has suggested that metformin might have various beneficial effects on aging-related processes. It is thought to influence cellular metabolism, reduce inflammation, target senescent cells, and improve mitochondrial function. Several clinical trials are underway to investigate metformin's potential anti-aging effects in humans. These studies aim to assess whether metformin can extend healthy lifespans, delay age-related diseases, and improve overall health in older individuals. It's yet to be approved by any regulatory authorities like the US Food and Drug Administration. It's been found that some people are predisposed to live longer than others, regardless of whether they're trying to or not. Many centenarians are quoted as having smoked, drank, and generally made no scientifically backed effort to live for as long as they have, with some citing things like peanut butter as the secret to their longevity. The truth is that most of these individuals have similarly long-lived family members, which led to studying their genetics for the keys to longevity. If scientists can find the genes that contribute to a long, healthy life, can they be given to those who don't have them? Some research teams are exploring genetic therapy as a means of combating aging. By using gene editing techniques like CRISPR, they aim to modify specific genes involved in aging processes, thereby extending healthy lifespans. Returning to the mice, we can see an example of genetic manipulation affecting aging. Typically, when a DNA break occurs, repair proteins will jump into action to restore the DNA before moving back to their starting positions. In the case of the altered mice, the epigenetic process of DNA break and repair was accelerated, with the placements of these more frequent breaks specifically chosen to avoid sections of the DNA that code for genes. As a result, the process didn't directly affect the genetics of the mice, but rather, by accelerating the break and repair process, caused the epigenetic elements of the altered cell to wear out and age. Proteins failed to return to their original positions, and the chromatin contained the DNA lost some of its cohesion. This caused the cells and affected mouse to noticeably age. This led researchers, including Harvard's David Sinclair, to the conclusion that epigenetics play a key role in aging, and that an epigenetic treatment could restore cells to a younger state. To test the theory, they reset the epigenetic markers in the affected cells to their original positions, and were able to safely reverse blindness and rejuvenate kidneys and muscle tissue in mice. Of course, there's more study to be done to determine if this can be done for humans. Stem cells have often been touted as a possible way to replace or restore damaged cells. To put it simply, stem cells are blank slate cells in our bodies that can turn into any other type of cell depending on what's needed, be it blood, skin, or organ tissue. We have fewer of them as we get older, so the question becomes, if we have more of them, can we stay younger? Unfortunately, it's not that simple. While scientists have found ways of creating stem cells from mature cells donated by adults, bypassing the controversial issue of needing fetal stem cells, the process is tricky. Resetting a mature cell back to an embryonic stem cell causes it to lose its identity, meaning that in many applications where it becomes a new cell, it might be harmful to the body due to mismatched identifiers, such as haplotypes of HLA. This is where another exciting avenue of research comes into play: Yamanaka factors. These are a set of protein transcription factors named for Nobel Prize-winning stem cell researcher Shinya Yamanaka, whose team discovered them. They play a vital role in transforming cells into induced pluripotent stem cells. iPSCs are a form of stem cell that maintain their identity while having the remarkable ability to differentiate into any other type of cell in the body, making them a powerful tool in rejuvenating aged cells. They can be made from various cells within the subject's own body, such as blood and skin, meaning they can easily be matched to the patient. These will likely also prove invaluable for growing organs for transplantation that are a genetic match to the recipient. By leveraging these Yamanaka factors, researchers are exploring ways to reverse the aging process at a cellular level. The goal is to turn back the biological clock and restore cells to a more youthful state, potentially alleviating age-related ailments and promoting overall health. Some studies have suggested using just 3 of the factors due to carcinogenic concerns regarding CMYC. This still allowed for cellular rejuvenation using the remaining factors and has been proposed as a safer method. While many approaches are focused on maintaining the health of bodily tissues at their present state, iPSCs could go much further in de-aging cells. While each group of researchers focuses on specific mechanisms and targets different aspects of aging, their collective efforts contribute to a greater understanding of the aging process and potential solutions for a healthier and more youthful future. However, amidst the promising strides in longevity research, it's crucial to be cautious and discerning. The field is flooded with pseudoscientific claims and so-called miracle cures, from diets to ice baths that prey on people's desire for eternal youth. Extreme procedures such as youth blood transfusions, despite radical claims, have shown no measurable benefits in humans in scientific studies. The FDA has actively advised against the treatment due to the lack of benefits and inherent risks of blood transfusions. As the journey of innovation continues, driven by the dedication of scientists, researchers, and institutions worldwide, we remain hopeful for the possibilities that lie ahead. Research into healthy aging aims to reduce the risks of cancer, heart disease, and mental decline. Lofty goals, but even small advancements will save and preserve the quality of human lives. Thank you for joining us in this exploration of the latest longevity research. We hope this has given you an overview of the current avenues of study in the field. Patents, intellectual property, and scientific innovation are of interest to you. Please subscribe for more exciting content.